Let's dive into a crazy paradox that's happening inside you right this second. Your body has trillions of cells. We're talking neurons firing, muscles contracting, but get this, from your brain all the way down to your big toe, almost every single one of those cells contains the exact same genetic blueprint, your DNA. It's a fantastic question, right? If the instruction manual is identical everywhere, how on earth do you get all this incredible variety? How does a single basic stem cell know it's supposed to become part of your eye and not, say, your heart? Well, the secret isn't in the blueprint itself, but in how that blueprint gets read. So, a good way to think about this is to imagine your DNA as a massive script for a play with thousands of different roles. A single cell can't possibly perform every part at once. It needs a director, someone to tell it which lines to read and which scenes to perform. And believe it or not, that's pretty much exactly what's happening inside our cells on a microscopic scale. These molecular directors are specialized proteins, and they're not just passively reading, they are active agents. They land on very specific sections of your DNA and start making decisions. They're the ones telling a cell, hey, you're a liver cell, so I need you to activate the detoxification genes. Or, okay, you're a neuron, fire up the genes for sending electrical signals. These directors are the absolute key to specialization. The technical name for these little directors is transcription factors, and they basically function as these powerful molecular switches. By latching onto the DNA, they can either flip a gene on, activating it, or flip it off, shutting it down. It's this simple on-off action, repeated millions of times all across your genome, that literally sculpts a generic stem cell into a highly specialized part of you. Okay, so this brings us to a huge logistical problem. The human genome is just vast. We're talking 3 billion chemical letters long. So how does one tiny protein, a single transcription factor, navigate this immense highway to find the exact gene it needs to switch on or off? It's like trying to find one specific house in an entire country with no addresses. Well, it turns out the directors don't go it alone. They have help. They have a navigation system, kind of like a cellular GPS that guides them right to their destination. And that system is run by a molecule you've definitely heard of, but maybe in a different context. RNA. Biochemist Yi Changlong came up with the perfect analogy for this. He said, if the transcription factor is the plane, then the RNA is the air traffic controller. It literally guides that protein to the exact landing strip it needs on the DNA. Without that guidance, the whole system would just be chaos. So you have this beautiful, elegant dance, right? You've got transcription factors acting as the directors, deciding which genes to turn on, and you've got RNA acting as the traffic controller, making sure they get to the right place to do it. It's this incredible partnership that lets a single do-it-all stem cell branch out and build all the complex tissues and organs that make you, you. But here's the kicker. What if we could hijack this process? This is where science gets really, and I mean really mind-bending. Researchers are no longer just watching this process happen. They're figuring out how to control it. They found a way to actually hit the rewind button on a cell's life, taking it all the way back to its youthful, undefined state. This is called cell reprogramming. I mean, it's a radical idea. You take a specialized adult cell, like a skin cell, and you tell it to forget its job, erase its identity, and shed all the age-related damage it's picked up over the years. The goal is to turn it back into what's called an induced pluripotent stem cell, or iPSC, which once again has the amazing potential to become any type of cell in the body. And to really get why this is such a game changer, we need to look at a devastating disease where the aging clock runs way, way too fast. Hutchinson-Gilford progeria syndrome is a rare genetic disorder where children age at a terrifying rate. It gives us a tragic but really powerful way to study the mechanisms of aging itself. Now, this diagram might look a little complicated, but the key thing to focus on is in that bottom red box. In progeria, a single genetic typo leads to the creation of this toxic, faulty protein called progerin. This gunk builds up inside the cell's command center, the nucleus, and causes massive DNA damage, which basically makes the cell get old incredibly fast. So that's the problem. But here's the question that's driving all this research. What if that damage caused by progerin isn't permanent? What if we could use what we know about cell identity to just wipe the slate clean and start over with a healthy cell? And the answer, incredibly, is yes. Take a look at the diagram up top. Using this cell reprogramming, scientists can take a damaged, aged cell, what's labeled here as a somatic cell, and actually guide it backward. They can force it to ditch its age characteristics and ultimately turn it into a healthy, youthful iPSC on the right. They can effectively erase the damage. So, how do they actually do it? Well, the huge breakthrough came with the discovery of 4 very specific transcription factors, which are now called Yamanaka factors after the scientist who won a Nobel Prize for finding them. It turns out by introducing just these 4 molecular directors into an adult cell, you can trigger the entire reprogramming process. You can rewind that cell's biological clock all the way back to 0. Now, this technology is unbelievably promising, not just for progeria, but for tackling all sorts of age-related diseases. But moving this from a petri dish in a lab to an actual therapy for living, breathing people, well, that comes with some pretty major challenges.
Yeah, it's not quite that simple. There are 3 big hurdles to clear. First off, fully reprogrammed stem cells can sometimes go haywire and form tumors. Second, how do you safely and efficiently deliver these factors to only the right cells inside a person? And third, and this is a big one, how do you make a cell younger without also giving it amnesia? You want your heart cell to act younger, not to completely forget that it's supposed to be a heart cell in the first place.
So this has led to a much more clever and nuanced approach called partial reprogramming. The idea here is not to rewind the clock all the way back to the very beginning. Instead, you just rewind it enough to clear out all that age-related junk and damage, but you stop before the cell loses its essential identity. It's rejuvenation without the amnesia. And the technology to actually pull this off is advancing at lightning speed. I mean, instead of just injecting the factors directly, scientists are now testing much safer delivery systems. They're packaging the instructions as mRNA inside tiny bubbles of fat or creating these special chemical cocktails that can do the job. And as you can see with the mouse here, these methods are already showing some incredible promise in living animals, actually extending the lifespan of mice that have this aging disease. And this, of course, brings us to the ultimate question. We've gone from just trying to understand the secret language of our cells to actively learning how to rewrite it. Resetting a cell's biological age is no longer science fiction. It's happening in labs right now. The real challenge for us is to figure out what that means. How do we safely and ethically use this incredible power to redefine what it means to grow old?